Okay, glad to be here. And thanks to Rich and the other organizers for putting together this exciting symposium. I'm going to do a little bit more nuts and bolts kind of talk than we've heard so far. What I want to tell you about is something that we all do every day, searching for a specific object that we want to find. And we do that hundreds of times a day. It's a very common, critical task that we need to perform, and involves many perceptual and cognitive mechanisms. And before I actually tell you about that, let me just point out the collaborators who have contributed to this project. So what I'm going to tell you about is, I guess, what we've been working on for about the last 15 years or so, working towards a really principled theory of visual search in arbitrary scenes, including natural scenes. And that's kind of where we're going. The key idea here is that starts with the fact that our perceptual systems are the result of evolution by natural selection. And what that means is that, in some sense, our perceptual and cognitive systems are designed to support the tasks we have to do to actually get around in the world. And so the philosophy in the lab is something like this. What we try to do is identify and characterize natural tasks, measure the relevant environmental properties and biological constraints. By relevant, I mean relevant to performing those tasks. And then we here's the modeling part, as we try to determine how to use those environmental properties and biological constraints, how to use that information to optimally perform the task. Once we do that, the whole idea of this final step of getting determining how to sort of optimally use the information is that gives us principled hypotheses for what might be going on in the nervous system, and then we test that in behavioral experiments or in physiology experiments. The idea, I guess, is that evolution has pushed things in the direction of optimality, and you're kind of better starting off with that kind of a model than you are just armchairing it. So the task that we're going to consider here are covert and overt search tasks. And this is just, we're not considering the completely general case. I'm going to tell you what we are considering. So we're considering the stimuli would be arbitrary grayscale, natural backgrounds or backgrounds, images, whatever kind. We have some known additive target, like this little Gabor target, added at some location in the image. There's a prior on whether the target's present or absent. There's a prior over possible target locations. In the covert search task, there's central fixation, and the stimulus duration is 1/4 of a second. The typical time when you're saccading around in the scene, that's the snapshot you get essentially to do a covert search behavior. And then in the overt search task, the stimulus would stay on until you make a response, but you're allowed to make eye movements. So the goals, of course, what are they? Identify whether the target's present or absent. If it's judged to be present, what's the location of the target? Goal in the covert search is just to maximize accuracy. That's what we're going to focus on. And in overt search, minimize the number of fixations that you make for a given error rate. Okay, so it's a concrete task. All right. So here's just a generic, model, description of the overt search task. Any, plausible model of visual search with eye movements would have these pieces in it. Input is an image, some kind of internal prior representation of where the target might be, and some internal representation of your own visual system, a model of your own visual system, if you like. That we represent here. We're just describing it as a detectability map, how detectable are targets at different locations in the scene. So an image comes in, it gets encoded. The D prime map and the prior, or your knowledge, is used to compute some posterior beliefs about where the target might be. Then you can make a decision based on those posterior beliefs. If the beliefs are high enough that you kind of know the answer, you make a response. Otherwise, you select another fixation, collect a new image, and so on. Okay, so that's just generic overt visual search. The special case of this is a covert search where you just simply don't get to make the eye movement, right? Display comes up, get a quarter of a second. So basically, overt search always includes covert search as a special case. But even when you have no location uncertainty, that is, the target's only appearing in just one location, and you know what it is every time, there's always intrinsic position uncertainty. And you can measure that easily in humans, and so basically all the time, even if you're trying to identify a target that can only appear in one possible location in the scene, it's still a search task, because you have intrinsic position uncertainty. So the logic of the approach is we're going to start with the simple case, this one, and then we're going to step back up to this one, and then step back up to that one, okay? So starting here, covert search with no extrinsic uncertainty. And so let's see where. So several questions we're going to ask about that. And here are the first two. What are the major factors in natural backgrounds that affect our ability to identify a target? And, what are the factors, and then how do they actually affect identification accuracy? So that's where we're going to start. 150 years of research with simple laboratory stimuli has shown that there are three major factors of backgrounds that seem to affect human ability to identify whether the target's there or not. The luminance of the background, the contrast of the background, and how similar the background is to the spatial structure of the target you're trying to detect. There's a huge literature on this. This is just a demo, but you can see the effects are big, right? If the same target is added in the two columns, obviously luminance has a big effect. If the luminance is the same on the two sides, but the contrast is higher, it's harder to see. If the contrast is the same and the luminance is the same on the two sides, if the spatial structure is similar to the target, then you can't detect it either, right? So this is just standard, well-known masking effects. And they're big, powerful effects. But what we don't know is, one, how do those really translate to the real world? And two, how do they actually combine, right? People usually study these things, have studied them kind of in isolation with simple stimuli, and haven't really looked at how the factors combine in the real world. So to get a handle on that, we used a technique that an approach we call constrained sampling. And I want to point out, this is kind of a fun technique, and it could be used in lots of other domains, not just the one I'm telling you about, so there's a little generality in this talk. The idea is that if you know, for whatever domain you're interested in, what some of the big dimensions are that we know from regular experiments are the important dimensions that affect performance. The idea is to then basically collect real signals, natural signals from the world, and then bin them along those dimensions so that you can kind of sort them in this way. So just to take this example that we're talking about right now, what we'd like to know is how the luminance of the background, the contrast of the background, and structural similarity to the target, how those affect performance in the real world. Well, one way to get at that is to just take calibrated natural images. We have, a couple thousand of them, and we just divide them up into millions of patches that are the size of whatever target we want to consider. All right, and so what I'm going to tell you about is either a Gabor target like this or a plaid target. But you could pick anything. Our interests are more in what the backgrounds are doing in their variation than the specific target. We're talking about searching for a known target in arbitrary background. So that's the idea. And so we just bin them all up. And these would be examples of patches of natural background that would be the size that would fall underneath a target. So we end up with millions of patches, and then we bin them along those dimensions. And so here's the luminance dimension, contrast dimension, similarity dimension. This is just a big histogram. There's no big story in here. The color is just telling you how many patches of natural scene fall into that bin. So there's nothing deep there. What I should maybe tell you is what similarity is. What we mean by similarity is it's a phase invariant similarity. So what that means is we get the amplitude spectrum of the target, the amplitude spectrum of the background, and we look at the cosine angle. It's the cosine similarity between those. And we do phase invariant because we know from the masking literature that, that the phase is relatively less important than the spatial frequency and orientation content of the background. Okay? So that's the idea. So what's the point here? Well, what you'd like to do to really see how these factors affect performance in the real world is you'd like to measure psychometric functions for every bin in the space. That would take a while, like 100 years or so. But the cool thing is, and this is often true in the major factors that affect the nervous system, the effects of all these dimensions are quite smooth. Luminance follows Weber's law. In the laboratory, that's a straight line. So what that means is you don't really have to measure this whole space. You can very sparsely sample it and interpolate and get the story for all possible natural backgrounds that occur, how these dimensions work. So you can see the power of that is quite good. So that's the idea. And so to get a handle on what might expect, that's always a good idea. What we try to do is get some handle about what's approximately the optimal performance you could do in this. And we can't really solve that exactly because natural backgrounds are complex. But if you're just looking for narrowband targets like these, if you're in white noise, if that's what your background is, we know that the best thing you can do is make a little receptive field or a template that's the shape of your target and just apply that to the image on every trial. And the response of that template, if it gets above a criterion, you say, yeah, the target's there. If it falls below, you say no. Okay, so it's straightforward. But if you have narrowband targets, even if the noise isn't white, it can be quite non-white, it's still optimal for narrowband targets. For more complex targets, there are other things to do. It's a little messier, but that's it. So you can just do that, and we can basically run this simple little template matching observer on every bin for both of these targets in the entire space. And so what do you get when you do that? Well, you apply this template to every natural image patch in a bin, and you get a distribution of responses. That's what the purple histogram is here. They're generally Gaussian like this. In fact, the average excess kurtosis across the whole space is 0. So there's no heavy tails here, for those of you who know about such things, because you're restricted to one bin. Okay, so, and if you define your template to be, you divide it by its own energy, then it's easy to show that, that your threshold of this thing, in other words, what amplitude of the target would be just detectable for this matched template observer would just be the standard deviation of the response distributions to the background. Right? That's it. Okay? It's a degenerate case. So what do you get? Well, you get this beautiful thing. This is the standard deviation of the response of the template matching observer, which is also its threshold for detecting these targets in natural backgrounds. And this is the whole space plotted here. So each panel is a different luminance bin area, different luminance. Each color on this is a different contrast of the background. And then the horizontal axis here is the cosine similarity between the structure of the background and the target. And then there's 2 symbols plotted on each one of these for the 2 different targets. Okay? And the cool thing here, kind of amazing, is that these straight lines are a very, very simple little model that says that the threshold for a template matching observer is just proportional to the product of the luminance times the contrast times the similarity. In other words, this is separable, multidimensional Weber's law is the description for detection of these kinds of simple targets in natural backgrounds. And these are just scene statistics. There's nothing in here. This doesn't have to happen. Similarity doesn't have to have a linear effect or anything, right? So there you go. So then you can bring it into the laboratory and say, okay, it's how we know what to expect if you're doing something close to optimum. what do people do? Well, this is an experiment where all we've done is just go along the, back up here, where we've just gone along the cardinal directions in the space. That's all this is. So this is for luminance, for the two targets, right? The two targets are the two rows. This is the luminance column. These are average of 3 human observers for the Pleiad and for this. For luminance, this is for contrast, this is for similarity. And what you can see is that humans also are linear. And the little black dots here, actually, those are taken from the natural scene statistics. These are the template matching observer's approach. Performance, except for one thing, the template matching observer performs a little better than we do. So there's an overall scale factor, but that's all. In other words, all the slope differences and so on are, and both targets are all predicted. Okay. This, of course, does not test the separability, which is clearly here. And so this is new data, but these are the 3 from the previous slide for this target. And then here's this is separability predictions, and there's the separability test data for the human subjects. So basically, overall, it looks like, for this space, human thresholds are proportional to the luminance times the contrast times the cosine similarity of the background underneath the target. Okay, well that's kind of, all right, great. That's something, and it's counting for, these are big threshold changes. But a cool thing about this paradigm is that you do not want to ever present the same background twice, because, right, you're trying to get a general answer of how's detectable or targets in the natural world, so we never present the same background twice. And each one of these bins is in fact filled with hundreds or thousands of different background patches, so we don't have to. But what that means is that we have lots and lots of responses from every subject in a particular bin where we've controlled the luminance, the contrast, and the similarity. So what you do is you're taking the dimensions we know from the literature are really important, you're compressing them all down and you're controlling them for every stimulus. Now you can mine the behavior of the subject in that bin to determine if there are other dimensions that we didn't know about that have a big effect on performance. So that's kind of the logic. And we discovered another one. Actually, it was by chance, but we understand it now quite well. And I won't take you through how we discovered it and everything, and we call it partial masking. And just to give you a sense of what's going on here, each row here is just, all the backgrounds there are from one single bin in the space. But you'll notice that these are hard to see, and as you go across here, it gets easier and easier. These are all hard to see. As you go across, it gets easier and easier and easier. What's going on here? It's really simple, conceptually. You're throwing a target down in the real world. Well, you know, in regular old psychophysics, you have a uniform field of texture or a uniform field of pattern or a uniform field of luminance, whatever it is, it's uniform over that, and you just throw the targets down and you measure. But in the real world, that's not what's going to happen. Sometimes the background is going to change underneath the target. Its structure is going to change. And what that means is that the target itself will only be partially masked by the background. It won't be completely. And there's a way to think about it is that's like when we look at occluding objects. If an object is partially occluded, we ignore the occluding thing and only pay attention to the features of the object that are visible. Well, the same thing happens here. It's a very nice way of studying that phenomenon. You basically have occlusion by masking. Some parts of the object are masked by the occlusion. Okay, so to get a handle on that, what's the right thing to do? What processing should you do? Well, a simple thing to do is just say, okay, let's back away from the natural images for a minute. Let's just go back to simple noise. So here's white noise, demonstrating that. So here we have a lion's head target that's added, in this case, to a background where the contrast of the noise is modulating from low to high. And another one, same lion's head is added here, where the contrast stays the same and is intermediate between those two. If I present a zillion trials like this, a template matching observer performs exactly the same on these two. Of course, you don't, because you don't pay any attention to the information here. You only take the information where you know the noise is low and the signal, you could detect the signal reliably, right? So it's basically partial masking by occlusion. What does an optimum detector do in this case? You can solve it. What you want to do, it's easy to show, that you want to use a template now that's reliability weighted. You want to have an estimate of what the standard deviation of the noise is at every pixel location, and you would just want to divide the template by that at each location. And now you're going to have a modified template that's essentially going to downweight this information and upweight this, okay? And you can prove that's optimal, and like that. And you can solve for what the performance is. Again, I'm not going to walk you through this, but basically the threshold of this reliability weighted template matching observer in modulated noise is just the average standard deviation across the whole image, that's sigma, divided by this thing that we call the partial masking factor. And it's just a locally and globally normalized template, and it's just the norm of that template, okay? And you don't have to know why, but there it is. And it's simple and it's direct. So that's the math. So we can say, well, okay, let's compare. We'll just do a white noise experiment on humans so we can see what they do. And so what we're going to do is we're going to modulate how much the contrast changes, uniform to strongly modulated. And we can also modulate the frequency of the modulation. Here again, uniform, higher and higher and higher frequency of the modulation over space, because that's what's going to happen in real scenes anyway. And so we can measure performance in a parametric experiment with these variations. You get 13 different conditions. Every one of those conditions, a simple template matching observer would perform identically the same. But of course, humans don't, as you guessed already that they wouldn't, right? Their performance is modulated very strongly. Again, a template matching observer would perform the same for all points in here. Humans don't. And there's some individual differences, but the same pattern is there. And if you, I guess that's not going to work either. I'm too much of a pointer guy. But we've averaged the subjects here, and now we've got the solid curves there, the predictions of that reliability weighted template matching observer, with one little caveat. In order to really apply this thing, right, in the real, when you don't know everything, you need to estimate what the actual noise is at a location. And so we've estimated the size over which humans are computing that reliability. And it's about 0.21 degrees. And that's what we use to fit these data. Okay? So it looks like that describes humans in this case. So what about the when we go to natural backgrounds, right? So so the dumb thing is just say, well, let's just suppose people are doing the same computation that they're doing here in white noise, right? They evolved for real scenes. We've measured what they're doing in white noise. It matches this model. So what do they do? Let's just try applying that. That's so we did. And so this is the same things I showed you before, right? These are all patches from the same bin. But what we did is these particular bins in the high dimensional, in the space, in three-dimensional space, these are bins that have lots and lots of background patches in them. And so we were able to bin those again into 20 bins. This is the 5th, 25th, and so on, percentiles of that. So you can see, again, you can see what's happening, right? When it's easy, it's when the target region is only partially masked. When it's hard, it's when the texture is uniform and it's uniformly masked. So what happens? What do people do? Well, not surprisingly, as you already guessed, they're going to perform better in the partial masking cases. Thresholds come down quite a long ways. The separation between these is just what we already predicted before, because that's just due to the luminance, contrast, and similarity of the overall background. That's what bin we're in, is shifting this up and down. The partial masking is responsible for this drop. And so if we just run the previous model, that's just a template matching observer on this, sure enough, we get that, right? It performs exactly the same on all these trials. This model that we already talked about before, that's based straight on scene statistics, it predicts the spacing between these curves quite well, but of course it doesn't get the effect, partial masking effect. If we now include the partial masking effect, which is another separable factor, we get this, and now it gets something like 93% of the variance that the subjects are showing in the experiment. So what we conclude from this is that human thresholds in natural backgrounds seem to be pretty well described by a product of 4 separable factors: luminance, contrast, similarity, and the partial masking factor. So that's the first 2 questions. And the next one is, well, if you're really in a search task and you're really doing it in the real world, well, the target can fall anywhere within your visual field. And so you're not going to get anywhere if you don't have understand a little bit about how the variation in processing across the visual field affects your ability to identify the target. So that's the next question we need to answer. And it turns out, some of you probably know this, the ganglion cells in your retina are the kind of major information bottleneck in your visual system, early one. There are a lot more photoreceptors, and there are a lot more cells in your primary visual cortex. Number of ganglion cells, that's sort of the bottleneck. And so what one can do is just go to the anatomy here and just look at the average in human eyes about what the spacing is between ganglion cells in the retina as you change eccentricities. You go away from the center of the fovea. And these are some data, anatomical data, and you can build a little model based on those. And you get something like this. So right where you're looking, your ganglion cells are highly dense, and then the spacing between ganglion cells increases as you move away. What we know from the psychophysical literature, and we've done it with natural backgrounds, and there's a whole literature showing that basically your sensitivity falls off inversely with the spacing. Because the spacing also increases linearly with eccentricity, so it's falling off inversely with eccentricity. So this is a standard formula that seems to describe how sensitivity falls off across the visual field. So you can put that in here as a 5th factor. And now you can sort of put it all together here and get some idea of what detection should be. So here's our natural background without a target. Here's the luminance at every location. Here's the RMS contrast at each location. Here is the similarity to this target at each location. Here is the partial masking factor at each location. And if you were fixating here, this is how the sensitivity of your visual system falls off away from that point. And then you can compute what the threshold would be, and I'm plotting here is what sensitivity 1 over threshold, and how that would be. So this is probably pretty good prediction of your performance. If I were to drop that target anywhere in this scene, this thing is telling you about what your threshold would be for any point in that image. And this is just based directly on natural scene statistics and this basic biological constraint about how resolution falls off. The cortex has got plenty of horsepower to use up whatever's there in those ganglion cells. So, last question. What about the intrinsic position uncertainty? How does that affect target identification accuracy? Well, Well, it turns out that if you're a template matching observer, we stumbled on a closed form expression approximation that's quite accurate for what the effects of uncertainty are on performance. So if this is your detectability, so-called D prime, without any uncertainty, then if this is your intrinsic uncertainty position uncertainty function, which will vary with retinal location, just like detectability, then this is what your detectability will be given that uncertainty. So it's, I don't know, I can tell you later about how we stumbled on this, but there it is, and it's quite accurate. And so we know something too about how this, from behavioral experiments, about how this works. And in particular, we have pretty darn good evidence that the uncertainty function is proportional to the ganglion cell spacing as well. The more the spacing between ganglion cells, the more your position uncertainty. That's the way it works, and it's roughly linear. This is, again, this is an experiment that I could talk to you later about if you want to hear about it, but this is the kind of the linear relationship. And it's consistent with lots of other things in the psychophysical literature about uncertainty about position in the periphery. So we're almost there to where we can build models of covert search. Here we've got this is the closed form expression that we want. If this is the D prime at location X and this is the uncertainty. But to really get a model of search, you actually can't just get thresholds, which is what I was showing you. We have to have a model of the detectability, the D prime, the signal to noise ratio. And we can get that from psychometric functions, what that shape is, but we need a couple more parameters. But these are the only parameters you'd need for any specific target. So there's just an overall scale factor. That's just, again, it won't, whoops, dang. There's the spacing that I didn't mention. I should have mentioned that. So for every different target, the effect of the fall off in sensitivity as you go away from the fovea depends on the target, right? Very fine detailed targets are going to fall off rapidly. Coarse targets are going to fall off gradually, but they all fall off with that same function, just with a different scale factor. And then finally, to get the shape of the psychometric function, the uncertainty affects it, but we know what the uncertainty is, and you need to actually put something in here to capture and that sort of accelerating D prime performance. And that's probably not due to uncertainty, because we've got that. It's probably like spiking nonlinearities or something in the nervous system. But in any event, this parameter and the uncertainty parameter have no effect on any of the threshold predictions I gave you before. They'll only affect the shape of the psychometric function. Okay, so we can estimate those parameters then in simple experiments. So you can measure a psychometric function in the fovea, say for your target, and you can measure psychometric functions as a function of eccentricity, right? This is threshold, or this is proportion correct as a function of eccentricity. And from these, you can pin the parameters down. But once you got them from that target, now you can throw that target on anything. Any scene at all, and this model will make a prediction for what your performance would be in the search tasks or whatever. Okay, so am I past? Okay, all right, let's push on then. So essentially what we have now is essentially a principled model of human detectability search with no extrinsic uncertainty, based on scene statistics and some basic fundamental biological constraints. To get to a covert search, we need to add the extrinsic uncertainty. And that's pretty straightforward. The dumbest, what we usually do is start with the optimum rule, which would be to get the max of the posterior distribution over the target locations, but you can add attention things in there too. But this is a starting place. I won't show you that, skip that experiment. For overt search, right, we'd still have whatever we did for the covert search, right? Then we have to consider fixation selection strategies and stopping rules for that. But I'll just jump to the fixation selection. We've looked at a lot of these, and These are all possible hypotheses that are interesting, but again, starting with the optimum ones, here are the best. We've been able to prove that at least in 1 over F noise and things like that, in fact, now it will be true in natural scenes too with the new model. This would be, one search strategy is to pick the location where your expected accuracy will be best after you make the eye movement. Another one is to pick the fixation location that minimizes the expected entropy over the posterior distribution, if you know what that means. And I'll skip that, and you can implement that. And we've discovered that humans actually behave more like these near optimum, or they have many qualitative properties that are like this. They fail in some ways too. All right, I'll quit. So, but here's the main Here's the main take-home points. We used a natural systems analysis to develop a theory of visual search from first principles. Based directly on natural scene statistics, we found that 4 separable factors predict identification performance in the fovea for additive targets in natural backgrounds. 2 biological factors, sampling density of the ganglion cells and intrinsic position uncertainty, explain the reduced accuracy in the visual periphery. And the general theory of visual search is then built upon this foundation by including optimal and suboptimal hypotheses for covert attention and overt fixation selection. Okay, thanks.